Another biology revision stream. So, before we get into this one, I want to quickly go over a couple of things from paper one, and I'm also going to give you guys a handful of pretty reasonable predictions, I think, for what's going to come up in paper two for AS biology. I know a lot of these streams blow up, they get like 100, 200 views. Guys, this is not GCSE revision, this is AS biology, okay? So, whatever you do, don't watch this if you're in year 12, year 11, because you're gonna have no idea what I'm waffling about, okay? So, a couple of predictions. So, in paper one, we had a lot of questions on a lot of different things, but weirdly enough, we did not get any questions on meiosis or genetic variation, except for like the last question. The last question was like a reading question, and it was about mutation. It was about mutation. And one of the questions was actually about how a mutation could lead to no, like, change, like a functional protein. So, possibly we won't get anything on genetic variation, but I bet you we'll get something on, homologous chromosomes, something like that. Protein synthesis. That did not come up, which I'm surprised because I spent hours revising that and it just didn't come up. So obviously that's transcription, translation, splicing, things like that. I honestly feel like I'm very confident with this topic. I don't know how much revision I need to do. I kind of know the step-by-step order of everything now in a lot of detail, but we can go over it again. Nothing on transpiration or translocation. Also, nothing on just generic microscope stuff. Nothing on studying cells, nothing like that came up. So this is my prediction. I also think we might get a sketch the cell question. And I'm guaranteeing you we're gonna get like a pretty weird, essay-ish question. Those are my predictions. So what I have for you guys today is I purchased a predicted paper. It was only like £1. It's from a relatively reliable source. The first predicted paper, to be honest, wasn't that good. The questions were worded weirdly. It was a little bit tedious, So, if there are questions here which I don't understand, or things we haven't learned, there's nothing I can do. So. Okay, and this is literally a question which came up in paper one. Okay. Complete the table by ticking features that are present in each group. Okay. So you got plants, animals, fungi, and bacteria. I'm not going to do the whole table. I'll just kind of summarize. So, First of all, I always do this with tables. There we go. Plants. Okay. So first of all, cell wall. Which has a cell wall? Well, obviously, fungi have a cell wall which is made out of, I believe, is it murine? Murien? I don't know how you pronounce it, but I'm pretty sure that's that. Hold on. I know bacteria have a cell wall made out of chitin, but is it the other way around? I think fungi have the chitin cell wall, bacteria have the okay. And then plants obviously have the cellular cell wall. Carbohydrates stored as glycogen, that's going to be in animals. I think it's only in animals. I believe it's like a storage for mammals only. DNA associated with proteins, that's only in eukaryotes. So that's going to be animals, plants. I don't think it's in fungi because fungi are like unicellular. They have nucleuses though, don't they? That's actually a tough one. I know bacteria don't have histones. Do fungi have histones? Let's Google. I'm pretty sure they do. So I'm gonna go pop fungi as DNA associated with protein. Okay. Let's check. Let's go all the way to the bottom. Oh! Okay, so I was correct about that. I was correct about this. I was correct about this. But I did not know that carbohydrates are stored as glycogen for fungi. I think that shows we have barely done fungi. We've barely done fungi. So to be completely honest, I don't think that was my fault. I genuinely the only thing we know about fungi is the unicellular. That's basically all we know. And the cell walls are made out of chitin, yeah, okay, so fungi have the chitin cell walls. It's bacteria with the murine cell walls. Okay. Cool. Viruses are another group of organisms. Explain why viruses are not classed with the other living organisms. So this is basically saying why are viruses classed as dead? And I know one reason for this is they can't reproduce on their own. They have to reproduce with a host. Rely on a host for replication. So I think that would be the right answer there. Cannot reproduce without a host organism. Acellular. Have we learned that word? We have not learned that word, apparently. Because I don't seem to recall it. What is acellular? Containing... Okay, yeah, containing no cells. I mean, that's kind of obvious. But okay. So 1 mark. So far, I've dropped 2 out of... So I have... Okay, we'll make a little tally. We'll make a little tally over here. Correct, incorrect. So far, on that first question, I would have gotten 2, because I didn't know about that glycogen. On this question, I would have got 1, because I didn't put acellular. But okay. But those are 2 words which I didn't know beforehand. So you know what? I'm happy with that. Warfarin is a poison that has been used to kill rats for many years. There are now large populations of rats that are resistant. Oh, explain the increase in the number of rats. Explain the increase? For 5 marks? I'm confused as to what this is asking exactly. This is obviously asking about being resistant. So is this talking about mutations or genetic variation? What is this asking? I'm going to guess genetic variation. So I'm going to say genetic variation in population of rats. This obviously means some rats more likely to survive poisoning. See, I'm thinking, is this wanting me to also mention mutation here? Is it wanting to say mutation leads to... We can put it. Mutation leads to genetic variation in population of rats, which means some rats more likely to survive poisoning. So they will... So more likely to reproduce, while rats... Oh, hold on, allele. We should definitely mention allele. Mutation leads to genetic variation to survive poisoning due to resistance allele. Maybe we should specify that. More likely to reproduce, while rats without allele die, which results in obviously a high population of us with the allele. So mutation leads to genetic variation in a population of rats. Some rats more likely to survive a poisoning due to a resistant allele, more likely to reproduce while rats without allele die, which results in a higher population of rats with the allele. That's my little hypothesis there. Let's check if we're right. There is variation due to mutation. Okay, so I put mutation leads to genetic variation. 2 marks. Nice. Those with the gene/allele are resistant to the poison. So I put due to resistant allele. So more likely to survive. So that is a mark. Yep. Pass on the allele. So I didn't put pass on. I put likely to reproduce. But results in a higher... So I think 4 marks though. The only thing I didn't specify was that they pass on the allele. But I think I mentioned that. I mentioned being likely to survive, results in a... That was a decent question, honestly. So, in a question where it's on about being resistant, it's mutation which leads to genetic variation, which then results in a new resistant allele. So I think honestly the thing which screwed me over was wording. I should have said genetic variation means that... Okay. That's fine though, that's fine. We're doing alright so far. I have not revised it since the exam properly, so I think this is a good start really. I think that's it for paper 1, or for question 1. Okay, here. Oh, this is about, Oh, okay, this one is about tissue fluid, which is something I've never done exam questions for. Explain the difference in hydrostatic pressure of the blood at the venual end of a capillary compared to the arterial end. Okay. So I think we should firstly refer to high hydrostatic pressure in arterial and due to ventricular contractions in heart. That is the reason, I'm pretty sure. So that's probably a mark. Why is there less hydrostatic pressure? Because components in the blood will diffuse out of the capillaries. So I guess components in blood plasma diffuse out of into tissue fluid down a pressure gradient. I'm going to put increases volume, but I don't know if that's right. So, slash decreases pressure. Okay, so I think that last marking point probably isn't right. So I'm intrigued here. Which results in, obviously, at the venual end, a lower hydrostatic pressure. So let's check what it was asking us to say there. Pressure decreases due to a loss of fluid / volume. Hydrostatic pressure is higher at arterial end due to ventricular contraction. Okay, so 1 mark for mentioning that. I knew that to begin. Due to a loss of fluid, so I said components in blood plasma diffuse out of capillaries down a pressure gradient. I didn't really need to go into that amount of detail. All it was asking for is there is a loss of fluid. Loss of fluid in the capillary. That's all it was asking. And then the pressure decreases. I put increases volume / decreases pressure. I shouldn't have talked about that. Okay. So I'm gonna, you know what I'm gonna do? I'm gonna give myself half a mark for that. Even though that's not a thing in the exam, I think I did get the right phrase. Hold on, I need to check this. What's going on? Alright, sorry about that. I'm gonna give myself half a mark because this whole section here, while it was technically right, I went into way too much detail. I said components in the blood diffuse out, which is technically right. They do diffuse out of capillaries. So I don't know actually, would that be a marking point? Ignore reference to lumen size. Accept movement of fluid from capillary to tissue fluid. I don't know then. Components in blood plasma diffuse out of capillaries into tissue fluid down a pressure gradient. That is almost the same as saying loss of fluid. I mean, I said blood plasma. I should have said I should have said fluid in blood. fluid in blood diffuse out of capillaries into tissue fluid, down pressure gradient, which increases volume / decreases pressure. I think, honestly, I'm going to give myself 2 marks for that. Not 3, because, well, technically, yeah, I was right. Technically, it would be a fully correct answer. I think due to slight miswording at the end, I don't think I would have gotten it all. I think an examiner would have looked at that and ignored some of my answer. Using the diagram, describe and explain how substances from blood plasma enter the tissue fluid at the arterial end. Ooh. I can hear something. For context, my ears like clogged up, so give me a sec. Jesus Christ, one of these days I'll be able to do a revision stream without interruptions. So, describe and explain. So, we'll draw a little mock version of the diagram here. This is the arterial end, that's the venular end. This is the tissue fluid. How are we going to stretch this? My god, I'm trying to clog out my ear. For context, when I do the fight nights, I wear earphones, and I think it's a combination of me just sweating and obviously wearing the earphone, and it clogs my ear up. And also, the position I sleep in, I often end up sleeping, kind of by accident, with my hand on my ear, which creates like a vacuum. So all this to say, my ear is incredibly clogged up. I can barely hear a thing, actually. So that's why I'm having a little bit of a hard time concentrating today, sorry. Using the diagram, describe and explain how substances from the blood plasma enter the tissue fluid at the arterial end of the capillary. Okay, so how do I go in? Right. Firstly, we're going to say high hydrostatic pressure at arterial end due to ventricular contractions. That's what you should always say when talking about hydrostatic pressure. Next, we're going to say components. Alright, I guess it's going to be blood plasma. I know it says substances of the blood. Small substances of the blood. Exits. Should we say diffuse? Diffuse out of capillary. Pause down a pressure gradient. So I guess we should also specify there lower hydrostatic pressure in tissue fluid. 4 marks. How substances from a blood plasma, and I guess we should say large substances do not enter tissue fluid. Capillary pause into tissue fluid. Let's have a look at the mark scheme, shall we? Okay. Diffusion from, okay, hydrostatic pressure in capillary higher than in tissue fluid. So, yes, because I also said a lower hydrostatic pressure, so yes, that is 1 mark. Just 1 mark there, okay. Capillary walls leaky? What? Well, what the fuck does that mean? Do not credit diffusion linked to pressure. So I said small substances of the blood diffuse out of capillary pores into tissue fluid down a pressure gradient. I guess I shouldn't have said that. I should have said from an area of high to low pressure. So, I'm going to give myself half a mark because I was almost right. Accept idea of small gaps between cells. So I did say diffusion, so that is a mark. Fluid plasma forced out of capillary or moves from higher pressure to lower pressure. Hold on, yeah, down a pressure gradient. So I shouldn't have said diffuse. Okay, hold on. So I should have said small substances of the blood move out of capillary from an area of high to low pressure. As the fluid moves out, glucose/oxygen/molecule. Okay, so 3 marks. So it says, emphasis here is on small molecules carried out as a result of mass flow of fluid, not diffusion. So a high hydrostatic pressure at the arterial end due to ventricular contractions. Okay, so maybe, okay. I'm going to kind of rewrite this. High hydrostatic pressure at the arterial end and a lower hydrostatic pressure in the tissue fluid forces small substances out of the blood, out of the capillary pores, into the tissue fluid from an area of high to low pressure via diffusion. But it says, do not credit diffusion linked to pressure. Diffusion from high concentration to low concentration, hydrostatic pressure, fluid from higher pressure. Oh, I'm so confused here. It wants me to talk about diffusion, but I can't link it to the one thing which diffusion's used. High hydrostatic pressure at arterial end. So hydrostatic pressure at arterial end is higher than the hydrostatic pressure in tissue fluid. This causes small molecules to move out of capillary pores via diffusion as what? This is the most confusing mark scheme I've ever read. It wants you to make reference to diffusion, but then you can't explain why. You can't link diffusion to the pressure gradient, even though that's exactly what it is. So either I drop a mark because I mentioned diffusion, or I drop a mark because I don't. I'm going to give myself 3 marks for that. I think that mark scheme is a little bit botched. I think that mark scheme's very badly written, okay? Okay. Another 3 marker here. Kwashiorkor, a form of malnutrition, arises from insufficient protein in the diet. It is commonly observed in children, and it triggers swelling in the abdomen and ankles. Okay, so that's clearly when tissue fluid can't be removed, I guess. Explain how the shortage of proteins results in a buildup of tissue fluid. How would a shortage of proteins lead to a... Will that be talking about how waste tissue fluid is not carried in the lymph? Is that talking about the lymphatic system and how maybe the components of that are proteins? I genuinely don't know. How the shortage of proteins... Okay, this is a tricky one. How does a shortage of proteins result in the buildup of tissue fluid? I mean, we could say, I don't know. Okay, I'm gonna go on a very big risk here. This could be completely wrong, but hold on. So we have our cells. So the cells are unable to produce, if we assume that this is our lymphatic cell or whatever. Okay, so capillaries put, the components of blood, so the tissue fluid is there, but there are no waste products created, no waste products from the cell, such as urea, which means that the tissue fluid is not forced into the cells. It remains outside of the cells. Maybe that's what it's asking about? Honestly, that's the first question I've been stuck on. Let's Google. Oh, it's on about water potential. How would you have interpreted that, though? Hold on. Water potential in capillary not as low, which means a lower pressure, so less water is absorbed by osmosis of the... What the fuck? No, I'm calling bullshit there, because how would you have interpreted that as an osmosis question? Nothing about that mentioned osmosis except for swelling. And I'll be real, I thought the swelling was due to a buildup of tissue fluid, not due to osmosis. I'm calling bullshit there. I'm calling that a scummy question. Because there was zero way I was going to interpret that. There was zero way I would have interpreted that in an exam. I mean, now I know. Now I understand. So, the water potential in the capillary is higher, which means less water is absorbed into the capillary. So, I mean, even that doesn't make sense, though. If the water potential is higher here, then I guess wouldn't that mean that it moves via osmosis into the tissue fluid? How would a lack of proteins do that, though? You know what? No, I'm completely nulling that question. I'm completely nulling that from my results, because we have never learned this. We've never learned about the involvement of proteins, and the question was literally about proteins. How was I meant to interpret that it was asking about water potential? How would have I inferred that water potential how would I have inferred from that question that water potential was higher in the capillaries? And how then would I inferred that that would have caused it? The mark scheme here makes zero sense too. If the water potential is higher in the capillary, less water absorbed into blood capillary, surely the answer should be that it would move via osmosis into the tissue fluid, resulting in a buildup? Oh no, hold on. I think that does make sense, yeah. Because what you're saying is then tissue fluid won't move back into the capillary. But I don't get it. That is one of the worst questions I've ever read because it makes barely any sense. How on earth would you have inferred that? Hold on, look. Arises from insufficient proteins. Explain how the shortage of proteins results in the buildup of tissue fluid leading to swelling. There was not a single mark point there about how a shortage of proteins. The only thing I can think of is in the capillaries there are proteins, so less of them would mean that there is, I guess, less volume in the capillaries. So then it would increase in size as water moves in from... How would it... I don't... No, I'm utterly calling bullshit. You know what I'll do? Because I paid good money for this, I will give myself 1 mark there. 2 wrong marks is what I'll give myself there. I'll give myself 2 bad marks because I guess I could have remembered that capillaries contained proteins, and I guess I could have somehow linked that to water, and I think swelling, looking back, would have connoted osmosis. But no, like, come on. That is a difficult question for 3 marks. I feel like if I asked anybody in my biology class, they would have gotten no marks. I feel like no one would have got that. So I'm going to give myself 2 dull marks there. Let's continue. Students wanted to investigate the rate of uptake of water from a plant shoot. A pedometer is a piece of apparatus that can measure the uptake of water. Okay. Explain why the students cut the shoot underwater. Oh, I've completely forgotten this practical. And we did it like the other day. I know you would cut it sideways to increase surface area. You would cut it underwater so that no air bubbles can form in the xylem, I'm pretty sure. Right? Yeah, I'm gonna say no air bubbles in xylem. I think these are all going to be small questions. Explain how the students could have used the apparatus to make repeat measurements and how this would have improved their data. No idea, to be honest. How the students could have used the apparatus to make repeat measurements. I think improving data because you get a more reliable mean. I think that's a reliable mean / average. I know that that's definitely a mark point. How they could have used the apparatus. Would this be something like... Would this be something like affecting the conditions of a plant to reduce / increase the rate of uptake? Or is this wanting you to... Or is this one about creating more air bubbles? I don't know what that's asking. That's a really vague question. The results are shown below. The diameter of a tube. I don't have a calculator, and I can't plot it because this is on a computer. The students repeated the experiment with a fan placed next to the plant shoot. Sketch a line. I can't do that. Okay, so a difference you would expect to see is higher wind means a higher transpiration rate, because water from the leaves is obviously blown off. So a higher transpiration rate means the water column moves up faster, which means a higher intake of water from the roots. Nasty ass questions. I'm gonna tell these guys to piss off right now. To prevent air entering the chute. So I said no air bubble in xylem. To prevent air entering chute. I guess I would have gotten that. I should have been a bit more specific. So no air enters the chute. I'm going to give myself half a mark for that, because I used some right words. Okay, use the reservoir to return the bubble to the start. Increases reliability of results. So I said reliable. Ignore references to accurate or precision. Okay, so again, I'm going to give myself half a mark for that. So far I've dropped half a mark and 1.5 marks. Because, yeah, I mean, that makes sense. If you want to... okay, no, genuinely, that does make sense. If you have the air bubble here at 10, yeah, you would return it to 0, but I don't recall how you would do that. You would use the reservoir to return the bubble to the start. Okay, I didn't know that. Increased rate of uptake. Yeah, that's what I said. Because... air lowers the concentration of water. Oh, my ear just went. Oh, that's so good. That's fine. Because air movement increases / maintains the gradient. Okay, so okay. That's annoying. So I would have gotten a mark, but then I would have lost one because I didn't actually specify. I didn't specify how. Yeah, sure, water does fall off the leaf, but what would that do? It maintains a concentration gradient, so a higher rate of transpiration. Okay, honestly, I'm still a bit salty after that first question, though. I think what we'll do is we'll do 2 more sections, and then I'm probably heading off. But I mean, we've been going for just about... state the genus of a clouded leopard. That's Neophelis. Nice easy question there. Okay. A student stated that all of the leopards must be closely related to each other. All of the leopards must be more closely related to the than the big cats because as they all have leopard in their name. Does it support their conclusion? No, because I mean, I guess you could specify the clouded leopard. They all share a common ancestor. But you could also say that the lion and jaguar share the closest common ancestor. You could say that the tiger. Yeah, you could say all of them share a common ancestor. You could say the lion and the jaguar share the closest common ancestor, meaning they're incorrect. The clouded leopard and snow leopard are loosely related. I don't know how you would word this though. This is a difficult question actually. So yeah, 1 mark there. So you would have gotten no. They all share a common ancestor, but they are not more closely related. The clouded leopard evolved first, so most closely related. Clouded leopard talking about the genus. The snow leopard is most closely related to the tiger. The leopard is more closely related. Okay, so I probably wouldn't have gotten a mark. I'll give myself half a mark just for the sake of convenience. But I get what that was on about now. If they have a different genus, obviously they are less closely related, or they can't be related. The snow leopard is more closely related to the tiger instead of the other leopards. I get what that was asking. It was just a little bit kind of difficult to interpret the diagram, but I get what it was asking. Okay. The snow leopard is quite different from all the other cats. Describe a physical adaptation of the snow leopard that would help it survive. This is on about surface area to volume ratio. So the idea is that basically a larger surface area to volume ratio means that it loses heat quicker. No, sorry. Okay, hold on. A small surface area to volume ratio means it loses heat quicker. So a large surface area to volume ratio means it maintains heat. So a physical adaptation to help it survive would be a larger surface area in order to maintain its temperature. Wait, what? Small surface area to volume ratio? Oh yeah, okay, no, I'm so silly. I got it the wrong way around. Because obviously humans have a small surface area to volume ratio because we maintain heat. A large surface area to volume means you lose... Okay, that was my bad, to be honest. That was my bad. That was a little mistake there. I knew it was going to be about surface area because we learn about it in that context, but okay. Unlike all the other cat species, the snow leopard cannot roar. Animal communication is important. Explain the importance of courtship behavior. The importance? I guess it helps distinguish between species due to their unique courtship behaviors. And it helps ensure, I guess, for survival, the reproduction. I don't know. What would you say for that? Genuinely curious now. Allows species recognition and increases chance of mating. Okay, so courtship behavior is species recognition. Allows organisms from the same species to... Honestly, I just want to count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So 15.5 out of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 15.5. So far we've gotten 50% of these wrong. Again, I did obviously give myself there 2 wrong marks due to that really badly worded question. And I think, honestly, I'm doing pretty bad at the whole species recognition. This is where I think I'm probably heading off, because you can tell it's kind of a downward trend. Ever since I got that question wrong there, it's been a downward trend of getting things wrong. This is a problem. The thing is with my revision is when I go through each question one at a time, do the question, do the mark scheme, I tend to start getting things wrong once I start getting things wrong. So look there, I got that wrong, so I started getting all the others wrong. So what I'm going to do, I'm going to take a break. I might be back later to do, I guess, more questions. Seems like the other ones are about biological molecules and ATP, which should be nice. Yeah, it's a question about ATP. Self-fractionation. Oh, antibodies. Cellulose. Testing for sugars. Microscope 5 marker. Phagocytes 5 marker. Okay, I definitely would have gotten the last couple of these right. Definitely. And to be fair, 50% isn't that bad, considering, again, for those unaware, 50% is approximately a B, and about 55 to 58% is an A. So all I would have needed to do there was clutch onto those 5 markers, those ATP questions, not overthink things. Notice how the questions about graphs screwed us over. The questions about all the recent things screwed us over. Like that practical, which I barely learned, came up, and I obviously dropped marks. Tissue fluid, I did quite well on that. The trees, which are just annoying as hell, but I get what it was on about now. I get what it was meaning. But anyways, I'm heading off. So I will see you guys in a bit.